So, as I was saying, there are many questions that are arising right now. First of all, from the beginning of the pandemic, we are asking ourselves, where did this coronavirus come from? And many people were pretending that it has been made in a lab and so on, whether it has escaped from a lab or done in purpose for some bioterrorism purpose. So, and the other question, of course, now that the vaccine is available, or the other many vaccines are available, if we should trust them, and what are the risks that we are taking if we get vaccinated, and who and when and why we should get vaccinated. And eventually, we would like to know when all of this is going to be over, and when we can get back to our normal lives from before. So I'm going to try to go with you over these questions and answer them one by one. So where did this coronavirus come from? Could it have been made in a lab? The answer to this is that most probably not, because obviously, if you want to make a new deadly virus, you start from the one which is the most similar to it. In reality, after investigation, it turned out that this coronavirus, the SARS-CoV-2, doesn't resemble the viruses that have been previously identified in labs. The one that it shares the most analogy with is about 95 or 90% homologous. While it is relatively common for new coronaviruses to suddenly infect humans, this didn't come as a surprise at all. We know that coronaviruses do this once in a while, and I'm going to show you why and how similar it is to wild coronaviruses. Actually, coronaviruses are a large family of viruses. And they infect humans, and they also infect animals. So these are zoonoses. And the particularity about them is that they have a predilection for their host, meaning that usually the ones that infect bats do not infect humans, the ones that infect camels do not infect other wild animals, etc. But accidents do happen once in a very long time. Until 2002, we only identified 4 human coronaviruses, and none of them was of medical interest because they only caused mild cold symptoms. So they were really not that serious. And then all of a sudden in 2002, we had bat coronaviruses that, one bat coronavirus actually, that crossed the barrier and infected humans. And this coronavirus was called SARS at that time, for severe acute respiratory syndrome. After 2 years, it disappeared. The epidemic was over. Only 8 years later, we had another bad coronavirus that infected humans, and this one emerged in the Gulf, in the Arabic Gulf, and it was called MERS, for Middle East Respiratory Syndrome. The respiratory syndromes were very similar, and it also turned out to originate from bats. We don't know whether it originated directly from bats or whether it passed a first barrier going to camels and then from the camels to the humans. What we know is that this coronavirus has actually indeed infected camels sometime before. So it is no surprise that the SARS-CoV-2 that is currently causing COVID-19 would be probably directly derived from a bad coronavirus as well. It could have been through another intermediate host, which is the pangolin, but we are still debating this fact. We don't know for a fact whether it went directly to humans or through the pangolin. What we know is that this virus here is 99% homologous to the SARS-CoV-2. So this is why we think that it is very probable that it derived in nature, from nature. So the normal question that we are asking ourselves then is, why did the other two disappear within a year or two, and why this one is causing a pandemic? And the question is simply because this virus is so easily transmitted by people who are asymptomatic. They don't have any symptom. And as we have seen, people went over New Year and they danced and had fun. And this is where the current flare is hitting us in Lebanon. So even though the fatality rate is much lower than that of the other viruses, it has been very effectively transmitted by asymptomatic people. And right now it is hitting 2.4 million total deaths around the world, and more than 100,000 people infected. So what we are asking ourselves is now how to prevent this virus infection through a vaccine. I will not go with you over the transmission and the methods of preventing the infection that everybody has already been over, but let's focus directly on the vaccine. To understand what are the different types of vaccines, I would like you to bear with me a little bit and see what is the virus made of. This is an RNA virus. It means that it is simply made of a molecule of RNA surrounded by a capsid, like all viruses, and around the capsid we have an envelope. The RNA encodes for all the components of the virus. It has the genetic program to make an entire virus, of course. The capsid has a role of protection. It is the internal structure. And the envelope is very similar to a cell membrane. And this is something which is quite important. I'm going to tell you why later. And in the envelope, you have the spike protein, the famous S protein or spike protein. So this spike protein is basically like a key that allows the virus to get into host cells. It is what we call the viral attachment protein. And it is also the one that is the most obvious on the surface of the virus. So it is the main target of the immune response, the immunity. So as you know, viruses cannot replicate on their own. In order to cause disease, they have to get into cells, infect those cells, and in the cells make new copies of themselves. So the viral infection cycle for SARS-CoV-2 starts when the virus binds to the receptor, which is the ACE2 receptor, which is expressed on a lot of different cells, but mainly on lung cells. This is why it causes pneumonia. Once the virus binds to the receptor, it gets into the cell, and then it releases its RNA, and the RNA starts making new copies of itself, and it starts encoding protein, meaning that it will tell the cell to start making all of its proteins. And the virus is going to hijack the cellular machinery. The cell will stop making its own proteins, and it will start making basically only viral proteins. Now we have all the elements to make new viral particles. And so the genome, the RNA copies get each into a new structure of capsid envelope, and we call this the assembly, and the virus gets released. So many different new viral particles will get released by the cell. So of course, when we have all of this that goes on, the immune system is going to prevent the virus from or defend itself to counteract the replication of the virus. And we have different ways the immune system can do that. First of all, the infected cells themselves are going to simply stop protein synthesis or slow down their protein synthesis And so they will slow down the production of new viral particles. But most importantly, after a few days, our adaptive immunity starts. And this is where we are going to have two types of immunity. The B cells, the B lymphocytes will start making antibodies, and the antibodies will bind to those new viral particles. And specifically, they will bind to the S protein, the spike protein, and cover it. So they will not allow it to bind to the receptor. So the virus cannot get into new cells. And so this will be a mechanism of defense. Even more important than this, we have the cellular immunity, where we have what we call the cytotoxic T cells. These T cells kill the infected cells. And so we have, of course, no new virus production. Why is the T cell response more important? Because with the antibodies, you are killing the product, you are targeting the product. With the T cells, you are targeting the manufacturing facility itself. You are killing the cells that make that product. So cellular immunity is very important when you are developing a vaccine. So let's see, what is a vaccine? And here I would like to stress on the actual definition, because some people are saying that these vaccines that we have, the RNA vaccines, are a device or they are gene therapy or whatever. This is not true, because by definition, a vaccine is an immunogenic, non-pathogenic preparation that is administered to stimulate the body's immune response against a specific infectious disease. So the purpose, any preparation that can prepare our immune response, prepare our immune system, and make it more capable of defending itself against a specific infectious disease is called a vaccine. So the purpose of the vaccine is simply to expose the immune system to components of the microbe. If they can make the components of the microbe or introduce the components of the microbe, then they fit the definition. And so if the immune system is being exposed to this microbe, it will not only fight it then, but it will remember those components. And the next time we encounter that microbe, then we can mount an immune response which is much faster and of a much higher amplitude. And so by being so fast in defending ourselves, we prevent the infection from spreading, and we prevent at least the complications of that infection. So we may be able to prevent entirely the infection, or we may not prevent the infection, but prevent the severity of the clinical signs. And at best, if we don't allow the infection absolutely not to take place, then we will not even be capable of passing this infection to somebody else and spreading the infection. Now, here I would like to stress that very few vaccines are capable of entirely shutting down the viral replication and the infection. But what we ask them at least is to prevent the symptoms. So what are the different types of vaccine preparations that you can have? Classically, people simply made what we call attenuated viruses. It means that they weakened the virus in the lab, made it less virulent, so it can infect cells, but it cannot cause disease. And then other forms of vaccines were developed where they were inactivated. So they were totally killed, and so they don't get into the cells. They get into the individual sees all their components, they can make antibodies against them, but they don't infect the cells. And when they don't infect the cells, unfortunately, they don't elicit a T cell response. We still make antibodies, but we don't make a T cell response. Another form is also through viral proteins, meaning that you don't take the virus, you just take the main target. In this case, for example, that would be the spike protein. And here come the new preparations that we are talking about, the RNA vaccine and the recombinant virus vector vaccine. The RNA vaccine is very simple, at least on paper. You simply take one small part of the RNA of the virus, only the one that encodes for the spike protein, and that piece of RNA is inserted into a lipid nanoparticle, basically a small fat sphere that resembles the cell membrane and resembles the envelope of the virus. So it allows the virus, the RNA, to get into cells. And finally, the recombinant virus vector is a way of making a vaccine where instead of using the actual virus, which may be dangerous, you take another one that serves as a vehicle to get you the RNA that you want into the cells of the vaccinated individual. So by driving the RNA of the virus, or by driving the genetic material of the virus, because we are going to see how it works, within the cells, it would allow those cells to make the spike protein and then the immune system to be exposed to it. I'm going to show you the mechanism in a while, but let's see what are the different COVID-19 vaccines that we are considering right now and how they compare. There are currently 6 vaccines that we are talking about. The Moderna and the Pfizer vaccine both relied on the mRNA technology. Both are extremely efficient, more than 94%. The problem with the Pfizer vaccine is that it needs ultra-low freezer, -70, which you do not find everywhere. It is simply present in some research and medical facilities. And then we have the Oxford AstraZeneca, the Johnson & Johnson, and the Gamaleya, or the Russian, or the Sputnik virus. All 3 are relying on the adenovirus vector technology. The AstraZeneca, the problem with it is that it is not recommended for people over 65 years, but it has a huge advantage, is that the company AstraZeneca promised not to make any profit out of it. So it is only $4 per dose, which means that a lot of countries can now afford it. Another advantage is that it doesn't even need a freezer. The Gamaleya needs a freezer, and we know that it is even more efficient than the AstraZeneca. And the Johnson & Johnson is not released yet, so we don't have to worry about it. And this leaves us with the Sinopharm, or the Chinese vaccine. This one, the phase 3 results are not disclosed yet. And it relies on a classic technology. It is simply the inactivated virus itself. So what do we expect from each one of these vaccines to do, and how safe they are? If I would start with the Sinopharm, since this one is the inactivated, the classic one, I would say that it's very safe. Of course, it's not infectious. However, it will not elicit a CTL response, and we don't know how good it is up until now. So let's see what are the other 2. A lot of people are saying, hey, how am I going to inject RNA into my body? Can the RNA vaccines change my DNA? This is genetic modification. We are going to turn into robots. Somebody even pretended 2 days ago, a cleric, somebody who's very respected in another country, that it may turn us all into sexually pervert people. Whatever, many fears. The fact is, all of this is not true. It's absolutely not based on scientific knowledge. Why? Because these viruses cannot change our DNA for the very simple reason that they don't even get into our nucleus. So when you inject an RNA vaccine, it gets into the muscle cells and it starts being translated, meaning that it gives the cell the instructions to make the protein. The protein gets exposed on the surface of the cell, and so it is recognized by the B cells to make antibodies. And again, this will also signal the T cells that this cell contains the protein, and so the T cells will turn into CTL, and this is what we want. This is very important. So by making antibodies and by keeping a memory, we get more prepared. And at the same time, it's very safe because it doesn't even get close to our DNA. Now, can DNA vaccines change my DNA? And again, the answer is no, absolutely not, because these vectors don't even replicate. These are replication defective adenoviruses. What is the principle? From that RNA that encodes the S protein, we make a DNA, we insert it into the DNA of the adenovirus. Now, there are many, many adenoviruses, and they don't even make symptoms, or at worst, they make a common cold when they are replicative. This one is replication defective. They don't do anything. So they get into the nucleus, but they don't get into the DNA. They remain separate. Then they make a copy of RNA, and the RNA makes the protein, and again, we get the same immune response. So as you can see, the technology doesn't bear the potential to change our DNA. Another fear is these vaccines have been developed too fast. And since it takes 10 years to develop a vaccine, these were available in 10 months. You see, there was a conspiracy. They were ready for them to take out and for the big pharma to make money. The fact is that this is not true. It is true that in classic approaches, it is true that it takes 10 to 15 years to make a vaccine. And as you can see, most of the time is taken for the preclinical testing. Vaccines go through different phases. In the preclinical testing, they are tested in a lab. Then a phase 1 is made, and then a phase 2 and a phase 3 for the clinical trials. The phase 1 enrolls a very limited number of people, about 10 people. Phase 2, about 50 people in a limited number of centers. And phase 3 enrolls even more people and involves a lot of money and is multi-centric. So all the preclinical trials were already made. Why were they already made? Because coronaviruses have been known for more than 50 years. And because we had SARS and MERS prior to COVID-19. So when SARS and MERS emerged, scientists rushed to make vaccines, and they were actually in the very late preclinical trials when the epidemics subsided by themselves. So they didn't put them in clinical trials. The adenovirus vector vaccines are also in advanced clinical trials for cancer, for HIV, for malaria. And so we know them in and out. And even RNA vaccines are in development, still are in development against SARS, but also against Zika and Ebola, which also made epidemics in recent years. So here we have all the platform which is ready and in which we only need to insert that small piece that encodes for the spike protein. The second reason is that we have an unprecedented worldwide collaboration. We have no budget limitations because we are facing a pandemic which is killing us all. No participants limitation. Everybody wants to get vaccinated, especially healthcare people, because they are at a very high risk. And there is a wide sharing of information and technology. So we are running from a competition environment to a collaborative environment. And because RNA vaccines, the ones that were released first, are so easy to manufacture. They don't require cell culture. They are simply pieces of a synthetic RNA. Now, the other question that people are asking, and they are raising a doubt, and they are saying that the companies themselves are halting the development of their vaccines, like we have heard for Sanofi. You see, they stopped it, so it must be dangerous. This is not true, because it is a normal process. Usually, this is the funnel we go through when we develop vaccines or any drugs. We go from basic research to drug discovery, to preclinical research, clinical research, and approved drug. And as you can see, every time to go from one step to another, basically 1 in 5 or 1 in 50 molecules make it through. So if we go to clinical research and they drop it, it means that it is a simple process, a true one. Finally, before I conclude with you, I don't want to take too much of your time. Can we trust these vaccines? And what are the risks we are taking? Here I took examples on the Pfizer, Gamaleya, and AstraZeneca vaccines. And as you can see, thousands of people have been vaccinated. By the way, I checked today that we have currently about, let me see the exact number. I think it is something like 5 million people getting vaccinated every day. We have reached almost 250 million people being vaccinated. And we have high protection. The threshold for protection required by the WHO, by the way, is 60. If you get above 60, you pass the test. And we have seen side effects. The side effects are moderate pain at the site of infection, headache, fatigue. And of course, there are people who are allergic, but the proportion of people who are allergic to the components of the vaccine is less than 0.5%. And this can be treated right away. And there has been no difference in vaccine side effects in the recipients and those who received the placebo, meaning those who received only the salt solution without the actual vaccine component in it. So who are the populations that should get vaccinated and why they should get vaccinated? A lot of people say, I'm young and healthy, so my risks are low. Why should I get vaccinated? First of all, we are shifting. We are seeing more and more people who are at risk of serious COVID-19. I'm sure a lot of you have heard about people in their 30s, in their 20s, who unfortunately died from COVID-19. And if you are vaccinated, you protect your loved ones. I'm going to get with you over this in a second, in the second slide. If you are elderly, of course, you are particularly at risk. If you are pregnant, we are not sure because this population has not been included in the controlled clinical trial, as well as those who are under 16 years old. The first one, because they are protected population, and the second, both of them protected population, and the second, under 16 years old, we don't see serious infection. Healthcare workers, of course. And if you have already contracted COVID-19, yes, there has been very few cases where people might get it again. So we think that the vaccine will reinforce the immunity. We simply have to wait for 1 or 2 months after recovery so that the vaccine would be effective. And for people who have chronic medical conditions, most of them are eligible, but they have to check with their doctors. So let me go over this idea of protecting the loved ones. We protect our loved ones through what we call herd immunity. How herd immunity works? Herd immunity means that if we have a large proportion of people in a population who are resistant to a certain infection, even if we don't reach 100%, This means that we can fight the pandemic. Why? Because if you have no one who has immunity around a patient, of course, the contagion will spread widely. If you have a few, it will have more difficult time. But if you have 70 and above, which is just a regular percentage that we consider on average, the spread of contagious disease is contained. So this is what we are aiming at. I don't know in Lebanon when we will reach it. خلوا يوصل بالأول الvaccine and let's have it available for everyone. But we can also expect to reach herd immunity by infection. Now, we have heard people who said that they are experts and they know that it is best to achieve herd immunity through infection. These people don't remember that, first of all, it's going to take an extremely long time with all the economic, the social, the psychological effect, the social effects, the educational burden, the burden on our healthcare system, or we can get it through vaccination. Now, we have to say that we don't know what are the long-term effects, but most of the adverse events in all vaccines happen within the first months, and we haven't seen it despite vaccinating millions of people. And the more early we vaccinate, the earlier we vaccinate, the less risk of virus mutation we are going to have, because the virus is running ahead of us and we are trying to catch it before it changes its life. So here we come to the variants. People are worried about the variants. A variant is basically a type of the virus which is a little different from the original one, because when a cell is infected with a virus and makes new copies, there are copying errors. Each one of these errors is called a mutation. And when the virus accumulates a certain number of mutations, it may acquire some characteristics which are not those of the original. So it may be actually better or it may be worse. The ones that are leading to lower infection are of no concern because they disappear. And those who spread faster or cause more serious diseases are the ones of concern. Right now we have three variants: the British variant, the Brazil variant, and the South African variant. It seems that the British variant is the one that is infecting us currently at a high speed in Lebanon. It is making the virus at the same time more contagious and 35% more deadly, according to more recent studies. The Brazil variant and the South African variant are becoming more resistant to some vaccines, but not to all. So this is why we don't know whether the vaccination is effective against all variants. We have already some data that the AstraZeneca vaccine is not effective against the South African variant. And definitely more variants are going to emerge if the virus is going to circulate freely. So prompt vaccination and early achievement of herd immunity are going to slow down this emergence. If we don't vaccinate promptly, we are going to have the scenario that we have with the influenza, meaning that every year we will have to catch up with the new variant. So in conclusion, should I get vaccinated? I hope I convinced you. My answer is certainly yes. Today I went to the hospital and it really felt to me like I was going through a no man's land during the war. I'm a person of the war, a survivor of the war, and I felt as scared as when I was running through a no man's land in the war. People has recorded so far more than 300 cases, and we have been through 3,500 deaths, and it doesn't seem to be stopping unless we vaccinate. of every 20, of every 100 test PCR, 20 are turning out to be positive. This is huge. And because our healthcare system is so overwhelmed, because some people left, some people are sick in the healthcare system, our case fatality rate has risen by 40% in 3 weeks. So we cannot keep going like this. We know that the vaccines are safe, we know they are effective, and we know that they are the best way to overcome the pandemic. So I hope you go, you get vaccinated, and you convince the ones around you. So I would like to thank you, and stay safe, everybody. Stay home. Thank you very much.
Thank you so much. You definitely convinced us of getting vaccinated. Definitely.
I hope so. And I'm sorry if I took too long.
No, Abad.
Can I ask you a question?
Sure.
Yeah. They often ask me, which vaccine are you going to take? I always answer, I've been answering that I say the first available vaccine. Am I right?
No, you are absolutely right. The first one you can get your hands on, go ahead and do it. Because for the vaccines to be released on the market, they have to be validated. They have to be approved by the FDA and by the WHO. We have right now 2 vaccines approved by the WHO, Moderna and Pfizer. No, I think if I'm, yes, both. There are, and all of them have proven to be effective and safe. The phase 3 have been released. We are waiting for the phase 3 of the Chinese, the only one which didn't get released.
Yes.
Thank you. Mr. Ghazi, you have a question? You raised your hand.
I would like first to thank Doctor for her beautiful presentation. And Doctor, I want to ask you about the AstraZeneca. We know it's a DNA injected in a chimpanzee virus that's harmful to people, and then transferred to people in a way. First, I want to ask you that, is it possible that with time, that virus that's transferred from a monkey into a human would probably affect the human? And second, it's a DNA that is managed inside the virus. Will it interact in any way with the human DNA?
No, actually, we have at least 15 to 20 years experience with adenoviruses being used as vectors for introducing DNA into cells. They have been used for gene therapy, and they are used, as I said, for vaccine development. So to answer your question, first of all, why did we choose a chimpanzee? Because not because it is safer than the human adenoviruses, but because when you inject the vector, the adenovirus vector into the humans, since we have encountered them before, the adenoviruses are very common, and so we are always infected by adenoviruses. Our immune system makes them less effective. So this is why AstraZeneca went for a chimpanzee adenovirus, hoping that it will not be immediately fought or attacked by our immune system. The Russians are using a prime boost regimen, meaning that they are using one adenovirus to prime for the first injection and another one for the second, so that the immune response mounted against the first will not attack the second. The DNA that is introduced by the adenovirus remains as a separate DNA and doesn't get incorporated into the cell genome. Actually, key parts of the DNA are removed, those that are important for replication, and those that might cause any interaction with the host DNA are removed.
Okay, thank you. Do you have new information about the AstraZeneca being injected into 2 steps, because they made a kind of error in the UK when they injected twice, I think, and then they got better
Yes.
results?
Yes, if you are interested, I found a very nice video about it. I could send it to you. What happened during the clinical trials in England, they injected by mistake half of the dose for the first dose. And the second one, people got the full dose. In other countries, they injected twice the 2 doses. And surprisingly, in England, those who got the first dose as half dose, they had a 90% efficiency. Those who got the full dose twice got 62% efficiency. So this is why you are seeing the discrepancy, the range of efficiency, and they are saying on average 70. So right now they are trying to figure out why by injecting less they got more efficiency. They don't know.
Thank you, doctor.
Thank you.
Mr. Wissam, did you have a question? You raised your hand.
Yeah. Actually, I wanted to ask you about the COVID-19, since you said that it's similar to MERS and SARS, but these two viruses are known as respiratory diseases, while COVID also attacks other systems in the body, such as the digestive system, and there are symptoms like the loss of smell and taste. So I just know what's the reason behind these new symptoms.
There is a mechanism of direct pathogenesis and there is a mechanism of indirect pathogenesis. The ACE2 receptor, which is the receptor for the virus to get into the cells, is actually expressed not only on lung cells, but also on heart cells, on cells in the digestive system, on endocytes, on endothelial cells, vascular endothelial cells. And actually it has been also seen in bladder cells as well. So this is why we are experiencing multi-organ infection. And there is another mechanism of indirect pathogenesis because it doesn't seem that the virus, that the viral load is entirely correlated with the severity of the disease. It seems that the severity of the disease is more due to the hyper inflammation that is ensuing infection and what we have been calling the cytokine storm. And it has not been what you see is that you have an exacerbation of infection at the time where the immune response is at the final phase of the innate phase, final part of the innate immunity, and the beginning of the adaptive immunity. It seems that it is the choice that the immune system makes to go in either one of its profiles, Th1 or Th2 profile, that is going to decide whether the person gets a severe infection or not. So yeah, it is behaving differently from SARS and MERS. All of them start as respiratory, but the complications we are seeing in COVID are very weird. I know somebody who had ocular complications.
Yes, thank you a lot. And concerning the vaccines, let's suppose that they are actually safe while they're made. How can we make sure that they are safe in Lebanon, actually, in terms of storage and delivering the doses to the people?
You mean in Lebanon when they get here, or in the lab, in the manufacturing?
Yeah, in Lebanon.
Sorry?
Yes, in Lebanon.
In Lebanon. Well, it happens that I know personally the person who is going to store them. They are going to be stored at Rafiq Hariri Hospital in the Center for the Influenza Surveillance. so in the center, in the lab of Rafiq Hariri, and they have freezers which are ready to receive them for the Pfizer. I know we are going to receive the Pfizer, of course, first. So the freezers are ready at -70. They will be distributed under the surveillance of people from Pfizer in trucks to the distribution centers, and there will be data loggers in the trucks within the boxes that are going to contain the vaccine. So I heard that. I trust it so far. Let's see. This is why it's a little tricky with Pfizer. The others are much easier to store. -20 is like a freezer at home. Really, every one of us can have a -20. It is no big deal. But -70 is more tricky, yes.
Thank you so much. Any other question?
Yes. I hope I convinced you.
Yes.
Doctor, a
Because
quick one. Now
we
that
are
you're, I
hearing
think,
a
in the middle
lot
of...
Yes.
Yes?
Okay, so I think these are all the questions, or am I hearing you well?
I
Yeah,
think
can
there was
I
a
have one more?
Yes, sure.
Okay, Doctor, how long do you think we will be able in Lebanon to vaccinate 25–40% of the society in order to somehow diminish the risk of contamination?
Well, there are different parameters involved here. First of all, how much are we going to be able to buy or to get? Because there is right now an international competition to get the vaccine. We have all heard about what's going on in Europe, that Pfizer made some promises and fell behind because they couldn't deliver everything that they promised to deliver. So it is going to depend on how much we can buy or get for free. It would be even better. And what is the rollout efficiency at the level of the government, but also at the level of the private institutions or outside the Ministry of Health. For example, at the Lebanese University, we are trying to get the vaccine in parallel with the Ministry of Health. Not the same, probably not the same vaccine. We don't know exactly which one we are going to get, and to vaccinate all the personnel and all the students and the families. So I think by doing this in parallel between the government and other institutions, or the Ministry of Health and other institutions, we will expedite it. The goal is to be all vaccinated before the end of the year, which is going to take some time. I doubt we will be able to achieve all this. This would be fantastic if we can. And a big burden, I think, a big hurdle in the way is the reluctance of people to get vaccinated. A recent poll is saying that we have only 30 people who are totally convinced that they need to get vaccinated. This is scary.
30 people?
30%.
30%. I heard that the army will be injected with the Chinese vaccine as opposed to the remaining of the population, which will get the Pfizer.
Really?
One more question, Doctor. Lately, Japan was suffering from lack of syringes because the syringe should take 6 units out of the bottle, while the Japanese syringe was able to take only 5. Do we have the appropriate syringes in Lebanon to use for this vaccine?
That I have no information about. I don't know. Honestly, I'm not personally involved in the vaccination committee or something like this. You probably know more than me about it. I have no idea about the syringes. Are they special syringes, you mean?
Yes, there are special syringes with a tipped head that would take all the 6 units out of the bottle, where in Japan lately they discovered that their syringes were able to only take 5 units out of the bottle, which will leave a little bit of the liquid, residues in the bottle that is not able to be sucked out. So
Ah.
I hope that here in Lebanon we will have the appropriate syringes to.
Hopefully. I'm sure the vaccination committee must be aware of that, but I have no idea whether they bought them or not. بكره بنشوف إذا رح يعملوا صفقة فيهم. I have no idea.
Probably.
The problem is that people don't have, it's in vaccination, a very major concern, a major parameter in success is the trust of the people in the government. And unfortunately, we don't trust our government. And this is why people are so afraid.
Yes, but,
But so far, I think the medical sector has proven a lot of, effectiveness and transparency, coping with the COVID as much as you can.
Yes. Yes.
Do you know how much it will cost to be vaccinated? Sorry to interrupt.
I have a indicative, very broad. Let me share with you again my screen. Can you see my screen?
Yes.
Okay. These are simply هيك approximate costs. For the AstraZeneca, it's only $4. While Pfizer is 20 and Gamaleya is 10.
So it's going to be $20 in Lebanon. This is the Lebanese.
Yes. This is what people told us after a meeting at the Ministry of Health.
Okay. So it's not going to be free.
No, hold on. No. Let me correct it. This is what they told us we would have to pay at the Lebanese university if we buy them. After a meeting with the Ministry of Health, the Ministry of Health is going to have, if I am not mistaken, a certain amount of vaccines free, and another amount will be a loan. We don't have to pay for it now. We will have to pay them we will have to pay for them later on.
If we survive.
If we survive. If we survive, exactly. If we survive, not from COVID, if we survive after the hunger كمان. هلا نحن we were considering the cost for the Lebanese university if we need to buy them at the Lebanese university, and we were very much tempted to buy the AstraZeneca لأنه it's the cheapest, it's the least expensive, $4. We don't have an idea about the Sinopharm. It is between $3 to $7, so on average كمان not too expensive. And the Russian would be a little more, $10. If you consider that we want to vaccinate all our personnel and the families and the students, this will make a huge difference.
Okay, thank you very much.
Thank you.
For the time being, the government is trying. Of course, it's going to be free, and maybe later on, even for all categories, it will be for free. We don't have to pay.
As a public, you mean?
Yes.
The government will make the vaccine free for people. I was talking about whether the government would pay.
Yeah, this is another business.
That's their business.
Yes, I was asking for the public.
I don't know. For the public, it will be free. The ones that are available through the Ministry of Health, from my understanding, it's free.
Yes.
Okay, any other question?
Yes, please. I have a question.
Yes, Layan.
Why do you recommend vaccinating individuals that were already infected with the virus? I mean, يعني, why to expect that the virus would create immunity, the vaccination, and the virus itself wouldn't?
No, we are not saying that the virus itself wouldn't. What we are seeing is that some people who got infected again. Okay.
Okay,
so definitely there is if you look at the rate of infection among people who have already had COVID-19 and those who are naive, definitely the rate of infection among those who got already infected is much lower. So the infection is providing immunity. But we don't know how much immunity it is providing, because there are still people who get infected.
But is it related to the duration of immunity? يعني is
the duration of immunity the duration of immunity anyway to coronavirus is low. يعني it is like for cold viruses and for influenza. هيدول you get infected with them every year, and this is what we are fearing right now, that we may
have to get vaccinated every every year. يعني we have to be vaccinated on yearly basis.
Yes, we might have to be vaccinated every year.
Could it be part of the vaccine of the influenza vaccine? يعني فيها تكون incorporated within هيدا الفاكسين نفسه?
Later on, maybe. I don't know. يعني هو when you want to make a combination vaccine, you have to run again your clinical trials as if it was a new molecule altogether.
Yes,
you go through all the phases of clinical trials again. It might be considered every time you have to vaccinate people, logistically it's difficult. It's logistically difficult to convince them to come and get their shots twice.
صح.
The more we combine, the easier it gets. يعني هلا بتشوفي كل childhood vaccines are combination vaccines. So let's hope we will not need it every year. تنشوف شو رح يصير.
Hopefully. Thank you so much.
Dr. Sulaiman, I think that when the virus spreads a lot, of course, it will be mutated and it will be more aggressive. That's why it's so important to get vaccinated so that the virus won't spread and we can control it when we are vaccinated. Yani it will be controlled. Yes, before the mutation will continue and it will be more and more aggressive. So this is very important that people know that we are going to be vaccinated so that it won't be aggressive and it won't spread. Already it's spreading a lot.
Yes, aggressive in terms of spreading. يعني it is going to spread and evade the immune system more and more, and we will see more variants. It is, and it reminds me a little bit of cancer. يعني when we have the early phases of a cancer, we target the cancer cells very efficiently, and as the cancer progresses, it evades the immune system more and more by mutating. The cells will accumulate mutations, and you end up having a very heterogeneous population of cells that the immune system cannot eliminate entirely. On the other hand, epidemics have been through history quite a bit, and they usually start and finish after a while, because the virus, when it gets into a new species and it adapts, it becomes less deadly. يعني the advantage that the virus has is not to kill the host, it is to live with the host so that it gives the host more time to spread it more efficiently to others. New variants may become less deadly, but they will be more transmissible.
They will.
So
Of course, the virus will not disappear. That's why we need to have vaccine
Absolutely.
every year, definitely.
Yes, this is where we have to interfere the earliest possible.
Exactly.
Okay, any more questions?
Sorry?
Any more questions? Okay, so let me again thank you, Dr. Suleima.
I
was
am the one who needs to thank you. It was really a pleasure discussing with
very
you.
informative. It was very clear. It's comprehensive, either for the scientific part or for the clinical part. I'm very happy. I'm very happy to be able to meet again at MUBS. I miss
Yes,
you.
inshallah, we will meet again live. We are getting all tired from meeting online.
I hope we'll have more collaboration with the Lebanese University.
Thank you. Thank you very much. It was my pleasure.
Thank you all for your attendance. Thank you all for your valuable question. Thank you, Dr. Joanne.
Thank you, Dr. Nina, and thank you, Dr. Suleima, for spreading scientific facts among all the false news we're being bombarded by. So thank you. I will make sure personally to share this, the live that was on Facebook, for the biggest number of people to see these facts, because unfortunately, as Dr. Nina already said, people are too afraid because they're not aware of the scientific facts about this.
Thank you. I would very much appreciate if I could get the link as well. So
yes, definitely. I'll send it to you by mail.
Thank you very much, Dr. Karam. And I would like to thank all those who asked me the questions and made this discussion a very fruitful one. Thank you again, Dr. Zaidan. I hope to see you soon.
Yes, me too.
Yalla, have a nice evening, everyone.
Thank you. Have a good evening.
Yalla, good night. Bye-bye.